Chapter 13. Oxidative stress, substances and genetics. And this is specific training for the Toxo Sensor genetic test. This test looks at three different categories. And the first is oxidative stress. You might have heard this word before. It's all mediated by free radicals. Free radicals are toxic chemicals that are produced by our metabolism, by conversion of oxygen. And this can, for one thing, damage the tissues and cause aging. They are one of the critical factors for aging of skin and everything. And they can also cause cancer. And if we look at the free radicals in cells, there's one very good example. Around 5% of oxygen that we use to convert energy is converted into a very potent free radical called superoxide. And this happens outside the cells, in the cell's cytoplasm, so inside of the cell. And the cells also have something like powerhouses. They convert energy and they're called mitochondria. And you also get superoxide produced in the mitochondria. Nature, however, found out that there's something happening there, that this is dangerous for cell health. And it gave us different genes that produce enzymes, and there's the SOD1 gene that converts superoxide to a different substance, to hydrogen peroxide inside of the cytoplasm. The SOD3 gene does the same thing, produces it outside cells, and mitochondria do it, or SOD2 does it in the mitochondria. Now, hydrogen peroxide is also a free radical, however that is then processed into other free radicals and then eventually it is neutralized. So this is a very critical protection that we have through these genetic variations. Now the problem is that quite a lot of people have a genetic variation in the SOD2 gene, which means that within the mitochondria the neutralization of superoxide is not going to happen. So if you look at the science, and this is actually a cutout from the report, that's the SOD2 gene, superoxide dismutase 2 in the mitochondria, and this here is the kind of like the catalog number for the genetic variation. As you know, we have three genotypes, CC on both genes, TT on both genes, or one gene has a C, the other one has a T in this genetic variation. And this is how common this is in the population. So 20% have CC, 53% have CT, and so on. And this is the result of this particular person. If you don't know much about this, I would recommend the training for simple disease risk statistics. That will explain it all to you. So CC, 20% have good protection against free radicals, 53% have only one active gene, so limited protection, and TT have actually two defective genes. And this is the science behind it. So this is the first aspect, the superoxide dismutase. Then there are some other genes that neutralize other free radicals. And there is the GPX1 gene. It's a selenoprotein, meaning that this protein is produced, but it needs selenium to become active. So the GPX1 gene has the instruction of how to build a small machine, an enzyme, and this enzyme requires selenium to be integrated, and then it can recognize free radicals and neutralize them. It's the general principle. So what we find is if people have selenium deficiency, they don't take in enough selenium, not enough of these proteins are activated, and they have low activity and low protection from these free radicals. Science has also shown if you then increase selenium by eating food that contains a lot of selenium or by supplements, you can increase the activity. So this is the case in normal people that have too little selenium. Now there is a genetic variation in the GPX1 gene that changes the blueprint for the protein. And what happens is that this protein is still produced, but it has a slight modification. And even when you integrate selenium, it can neutralize free radicals. However, binding or its function is less effective. Let's say, for example, 50% less effective. This means that even when you have normal levels of selenium, you get low levels of activity. And science has shown that if you increase selenium even higher, you get more activity, and you can kind of rescue the protection from free radicals if you have the genetic variation by having higher levels of selenium. And this here is the gene, GPX1, glutathione peroxidase. This is the genetic variation code again. This is what the gene does. And you see 67% have good protection, but 26% one of the genes is modified, and in 7% both genes have this genetic variation, so you have lower protection. And this here again is the science. And you can see there's a lot of science going on here. It's something science has known for many years. Then the next factor for oxidative stress, you might have realized that beauty creams or beauty products often contain coenzyme Q10, because coenzyme Q10 in our body can be modified, activated, and then is a very good protection against free radicals, which I said are one integral part of aging, and it is meant to reduce aging. So some people take coenzyme Q10, which is very expensive, but they take it as a supplement in addition. And coenzyme Q10, when it reaches our body, has no effect. It first needs to be activated by an enzyme, again, an enzyme like scissors. They cut open the coenzyme Q10, convert it to ubiquinol, and ubiquinol is the active form and has a strong antioxidant effect. And it can grab onto free radicals and neutralize them. So it is indeed a good protection against free radicals. This here is the gene that produces, you see this gene here. This is what it does, and 66% have a normal activity, so they can do this conversion. 30% have a reduced activity, so only one of the two genes does its work, so you can expect about half of the activity. And 4% of the population, TT genotype, they don't benefit from it at all, because the coenzyme Q10 cannot be converted to ubiquinol, and even though people take this expensive supplement, they cannot convert it into the protective effect that they are hoping to get from it. So what is happening in these people, you get the genetic variation, the coenzyme Q10 cannot be converted, and free radicals are not neutralized. So one function, or one thing you could do, is actually, instead of taking coenzyme Q10 only if this gene has this genetic variation, you can take the active ubiquinol that doesn't need to be converted anymore, and that can then neutralize the free radicals. Or alternatively, there are other free radical inhibitors like vitamin E, vitamin C, alpha lipoic acid. They have a similar role, and they can also neutralize free radicals. Okay, and this here is a cutout from the oxidative stress part of the Toxo Sensor. So you get a summary. You have a slightly elevated level of oxidative stress. So here you see your oxidative stress in cells. It can be normal or it can be very high, depending on how many of the protective genes have a detrimental genetic variation. This person can activate coenzyme Q10 to ubiquinol. So here we are in the possible part. It could also be not possible. So coenzyme Q10 is a recommended antioxidant. Otherwise, you would only be left with vitamin C, EA, or ubiquinol. So coenzyme Q10 here is absolutely functional. Also, the daily requirement of selenium is increased because we need more selenium to activate the GPX1 gene that I've explained to you. And the recommended dose of antioxidants is slightly higher than normal. So this is a cutout. This is the information that you get in the report. And then if you have an increased antioxidant risk, you get prevention advice. It's written in very simple language, so you can read through it once to find out what kind of recommendations we make. One is an antioxidant-rich diet. You could either do it by modifying your diet, which the nutrition sensor will help you to change your diet to get more free radical inhibitors, or alternatively, or in addition, it's possible to take supplements that contain these antioxidants. And we have one product, which is the NutriMe Nutrition, where we actually dose each of these antioxidants based on your genetics, so to make sure that you get the right antioxidants in the right dosages. So some key antioxidants are vitamin C, E, beta-carotene, alpha-lipoic acid, then either coenzyme Q10 or ubiquinol. As you remember, it's either one or the other. And selenium again is dependent on how much is required. And actually, there's one interesting point. Some people say, why do you do a genetic test? How much selenium you need? Why not do a blood test? And the problem is, irrespective of the blood test, you would say the selenium level in blood should be a certain amount, because you say this is the healthy area. So you would try to adapt everyone to be in the right area, and then you would say this is now in the right amount. But from science, we've seen that some people require a much higher amount because of their low GPX1 activity. So if you just normalize them to the normal area, you would actually not reach the right amount that you should get for this person. So the blood test would again try to put everyone in the same basket, saying everyone needs the same amount of selenium in the blood, but we have seen that this is not necessarily correct. And then also 2 cups of coffee per day. Coffee has a lot of antioxidants, and science has shown that in women that drink 2 or more cups of coffee and have a certain genetic variation do benefit from a delayed occurrence of breast cancer. And this is 7 years later on average. So really some women do really benefit from coffee drinking, while others do not. Okay, and then the next thing, this was antioxidative stress or oxidative stress. Then there are the substances that affect our body in different ways. For example, alcohol dependence. There are people which have a genetic tendency to become more alcohol dependent, while others have a protection from it. And here's the results. This again is how you would see it in a report. Your risk of alcohol dependence might be normal, or it might be increased. If it is increased, you should be aware of the increased risk, and you should reduce alcohol and maybe avoid it altogether to reduce your risk of developing a dependence. Then the question, how quickly is caffeine broken down? You know, there's some people that say they can drink a coffee before they go to bed, and others can't sleep anymore if they drink a coffee in the afternoon. So here we see how fast your caffeine is broken down. Then the use of cannabis has been shown to increase the risk of schizophrenia if the use was before the age of 16 and a certain genetic variation was present. So some teenagers out there are consuming cannabis, they have a certain genetic variation, and by combining these two in adolescence, they smoke cannabis. This increases the risk of schizophrenia later in life by 11-fold, so it's a significant risk. Some people, or most people, will already be past this age. In this case, it might not be interesting, but it might be an understanding if schizophrenia is present that this might have played a role. And then the effect of coffee and breast cancer can either reduce or delay the onset, or it cannot have any effect on the onset, and then it can be positive or not to drink coffee. And these are the 2 genes that are relevant for these, the COMT gene and the CYP1A2 gene. If you don't know what all of this means, please do watch the simple disease risk statistics. That explains the whole concept very, very well. So if we look at the COMT gene, this is a cutout from the report. This is how we can see it in the report. This explains what it does. And the AA genotype says no effect, but increased risk of alcoholism. The AG genotype, 55% of the general population have it. Increased risk of schizophrenia when cannabis is consumed before the age of 16 years. That's a 2.5 times higher risk. But a normal risk of alcoholism, and GG is a 10.9-fold higher risk. And this is the science behind it, in case you want to check up of what we claim here. And this is the end of chapter number 30, Oxidative Stress, Substances and Genetics, as specific training for the Toxo Sensor.